So we thought um, it would be helpful if you could briefly explain the vaccination review process in lay terms that someone who isn't in the field could understand the rigor and all the different levels that go into the vaccine review and approval process.
Yeah, so I'll do that, but I'll kind of highlight some of the differences that have happened with COVID, because I've heard that a lot, and that is a common misconception that these are experimental type vaccines, and that people don't want to be guinea pigs. That those type of comments I've been hearing, but the thing is, nobody is. If anybody is a guinea pig, it was the people that were in the clinical trials. So these vaccines have been tested in clinical trials, in the same kind of clinical trials that would happen for any other vaccine. The difference that has happened in this case is that one is that these weren't they didn't start from scratch. So a lot of the vaccines that have come to fruition right now, and there was a lot that came into us as clinical trial applications that were in very, very early stages, and we sadly had to refuse quite a few of those because they were just not quite there yet. But the ones that did come in that have come to market now, they had leveraged so much data from platforms that already existed. So either like Moderna had already worked on mRNA technology for several years, over 5 years, or there were products that had leveraged other platforms from Ebola platforms for like the AstraZeneca product and the Janssen product. They'd leveraged other um, products that they'd used with the same type of platform, or had leveraged other information from the other SARS information that they had gathered. They had started doing research on SARS and MERS, which are very similar viruses, but then those pandemics, or it never kind of came to what they thought it would be. So some of those products weren't and didn't end up getting marketed, but that research was all being able to be leveraged in terms of a non-clinical data, proof of concept, safety data that is looked at from a toxicology perspective. So they were able to use that data, and that saved them time. So that was one kind of time saving aspect. Another time saving aspect that they did in terms of this pandemic, and would not normally happen, is that the trials were adaptive trials. So that meant that they were doing things in parallel. So sometimes the phase 1 trials were conducted if they thought that there was enough safety information from platform data, and some of the challenge data that was done in primates, which took longer and was harder to access because everyone was accessing the same primates and same material, were done in parallel before the phase 2 trial. So that's some of the differences. And then some of the differences too was that some of the phase 1, phase 2, phase 3 were done in stages that they moved into each other based on information that they got from initial parts of the trial, and then they were moved on at a quicker pace to the next part of the trial. But nothing was actually skipped. So all of that was done. It's just that some of it was done in parallel. And the other big difference was that we were reviewing in parallel, which we don't normally do. So we were reviewing the data as it came in, as opposed to waiting till the whole trial was finished. We were evaluating interim data and making a decision based on that. And that's why the approval is not a full approval, it's an emergency approval, because we're still getting data in, and we have what you call terms and conditions based on that emergency approval. So if any data continues to come in that doesn't comply, or some other issue arises from longer term follow-up, then we have measures in place that we can revoke that emergency license. So that is some of the differences with how the process normally is and how the process is now. So the only difference in terms of is that the trials weren't completely finished. So there's still long-term follow-up that is ongoing, and that normally would have been completed before we would review a clinical trial submission. So I guess that's one difference. But for most vaccines, any adverse events that happen would happen usually within the first 48 to 72 hours. But more within the first 2 or 3 months. So that was the cutoff for the data that we wanted to see. And the long-term follow-up is still happening, but we gave this emergency approval before that long-term data became available. So some of that long-term data also includes how long the protection lasts for. So that data would feed into, maybe you'll need another booster. Maybe you'll need, maybe it won't last for as long as you think, but it will still help in term for managing this crisis.
That's a very interesting answer. I mean, it's so some of it is that they had the product sort of on the shelf, pop in your new antigen, the new thing that the community is being raised to, and that speeds things up greatly. Some of it is on your end in Health Canada, where you do things in parallel, like a computer works nowadays, as opposed to sequentially one after the other. You do it in parallel because there is a crisis that we have to deal with, and related to the depth of the review. So if a company like Pfizer wants their vaccine approved, do they give you a bottle, you check a bottle and you determine it is what they say it is, kind of thing? Or what's the actual level of chemical analysis of vaccines that actually come into the country? Are they spot sampled?
We do get samples that come in. Most of it, though, we're evaluating like paper review, in that they have to have a certain number of consistency lots, and we review all of the test results for those lots. And it's also to ensure that they have the right tests in place to catch anything that could go wrong. So things have gone wrong because things are happening at an accelerated speed in facilities all over the world. But if it is detected, that's a good thing, because that means that they have the measures in place to catch things that could go wrong. And the things that do, when they do catch it, nothing is put out onto the market. So that it's being caught before it goes out onto the market. So that's a large part of our role, to make sure that those measures are in place. And then we also do get samples in that we more test to ensure that, one, that we understand their testing procedure, and that it makes sense, that we can identify any issues with it when you do it yourself as well, and that we can spot check samples that come in that are going out for the market. And then there's also an inspection role too. So we don't. That's another arm of the government that also does an inspection of the facility. And there's also a more rigorous role for the country that is where the manufacturing facility is in place, that they do more oversight on the GMP of the facility, which is the good manufacturing practices.
And in fact, that latter thing you mentioned, good manufacturing practices, that's how they presumably caught that J&J problem in the States.
Exactly.
Right?
Exactly. So that was caught from the inspection stage, but from our stage, we had made sure that there was appropriate identity test in place, so that you could identify between the two products, and that one, would be able to discriminate against if it was a Janssen product or the AstraZeneca product. And that's because we knew that the products were being produced in the same room. We also made sure that there was, carryover procedures were in place, that there was appropriate cleaning validation. So that's what we can ensure for our end, and that there's testing in place to, ensure that there wasn't sterility issues, bioburden issues. But whether that's being complied with, we could only tell from the test results. But whether there's breakdown in GMP, that's what they can tell from the inspection side. But either way, it would be caught from either side.
Yeah. So it's a partnership with the manufacturing country and Health Canada.
Exactly. Well, Health Canada also does an inspection, but there's a bigger emphasis to do, for the local.
So